Yeah, good morning, everyone. Yeah, good morning, Sayed. Good morning, Evan. Good morning, Puneet. Yeah, first of all, I welcome you all. This is my first class with you people. I am Dr. Pooja Johari, and I will be taking your biochemistry classes. So I can see there are so many queries here. BDS also has the same subject. Yes, biochemistry is better there in all the medical streams, be it MBBS, be it BDS, be it physiotherapy, be it any subject, be it nursing also. Yes, biochemistry is taught in the first prof of every medical stream. And this is such a subject which will be very helpful for you in all the other subjects, relative subjects also. Okay, so let me first of all introduce to you what do you understand by this subject also. You should be able to realize the importance of this biochemistry. Right, so this is, yeah, so this is for first prof students, this is for students who have already completed their MBBS, those who are doing their internship, those who are preparing for this PG exam, for their FMGE exam, everything. Clear? So basically, what is this biochemistry? It is simply the chemistry of the biomolecules. Right. So what are these biomolecules? Molecules which are actually important biologically, right? Which include what? Which include the carbohydrates, which includes the fats, includes the proteins. So these three are the basic components of our diet, isn't it? What all we have? First of all, yes, is my voice audible to all of you and my screen visible to all of you? This is the first question which I should actually ask you before starting up with the class. Yeah, good morning, everyone. Good morning, Sayed. So is my screen visible, beta, to all of you? And my video clear to all of you? Yeah, good morning, Dilkhush. No, you can't use doctor in MBBS first year. There's a long way to go, beta. Yeah. But yes, inshallah, you will be able to put doctor soon. Okay. So basically, we have carbohydrates, we have fats, we have proteins in our diet. These are the three things which we have. Okay. Along with, we have vitamins and we have minerals also. Right. So now see, in biochemistry, what we are going to study, why we have carbohydrates in our diet, why do we have, how much we need to have, right? And what happens when we have eat carbohydrates. Okay, same is the case with fats. Why we require fats in our diet, how much fats which we should eat in our daily life, or how much quantity of fats we require in a day. Okay, similarly proteins. So these are the three components which how much we have to eat, what happens when they go inside the body. So everything we are going to cover, and along with vitamins and minerals, right? And second component of biochemistry will include molecular biology, which is again a very important concept. Molecular biology, study of the molecules which include DNA, which include RNA, and which include along with this how the proteins, because this is all related also. Just as suppose if some students of you must be entering in first year, so you have that kind of curiosity why we are studying medicine, what is the importance of medicine in our daily life. In our daily life also it's very very important. Just say if I talk about the concept of diet, isn't it? Right. So how much you have to eat? Okay, ऐसा नहीं है कि suppose if you start eating out under stress, you eat so many things, it will create a bad effect on your body. Okay, if you are eating little of it, you will be malnourished, you will be weak, right? And always remember, whenever we are eating little of the food, we are eating more of the food, it will lead to diseases. And some diseases have a molecular basis of inheritance. They are inherited as such. So that we are going to study in molecular biology. So this all we will be covering up with you people. Okay? Yeah, hello Dilkush. Yeah, how are you? Right. I will be starting with the basics. So don't worry. So those students who have already covered this subject, I mean those who are doing their internship also, they will also find this lecture very interesting. Why interesting? Because I'm starting from the basics. I will be teaching you entire core depth of biochemistry. So that if you understand this subject, you will see when you go in second prof, pharmacology is based on this. When you go in your third prof, medicine is based on this. So automatically you'll be able to correlate it, right? So like SPA. Okay, right. And whatever queries you have in your mind, you people can ask me. So to begin with, we will be starting up with the basic ones, carbohydrates, right? We all know what are carbohydrates. In a layman's language, I can say that carbohydrates are simply the compounds which have carbon, hydrogen and oxygen in the ratio 1 is to 2 is to 1. Right? Or you can say they are the hydrates of carbon. Clear? This is what they are, the hydrates of carbon. Now see, carbohydrates, the widely accepted definition is that they are simply polyhydroxy derivatives of Yes, I'll tell you the name also of the book which you people have to follow. Dilkush, see. First of all, my lecture is more than enough. But yes, if you talk about the books, a very good book of biochemistry is Vasudevan. D.M. Vasudevan is an excellent book. Then you can go for Satyanarayana. It's also a good book. It also covers the entire syllabus. And if you want to go for extensive one, you can go for Harper. Harper, the 32nd edition. It's like the Bible of biochemistry. Okay. So they are the polyhydroxy derivatives of aldehydes and ketones. And one very important thing which I want to just tell you before beginning of the lecture with you, if you try to understand biochemistry as keeping chemistry as the basics, you will find it very interesting. Because all of you have studied chemistry in your class 11th and 12th, so it will be very easier for you to understand. Okay, yes, very good, Sayed. They are the polyhydroxy derivatives of what? Aldehydes and ketones. And on hydrolysis, they will give aldehydes and ketones only. They will give aldehydes and ketones. Right. So if I'm talking about the definition of this, I can simply say that carbohydrates can be categorized depending upon the functional groups into two types. So depending upon the functional group, we all know that functional group is always given preference. So depending upon the functional group, they are categorized into two: aldoses. Aldoses, then aldehyde is the functional group, and if it is ketosis, ketone is the functional group. Right? And the most important thing about here is aldehydes and ketones are linked as isomers with respect to one another. This is the most important feature of carbohydrates. Because whatever we are having in our diet, if we are having something, aisa to nahi hai, we are saying you go and tell your mama to give you glucose. You never go and tell your mama to give you fructose in diet. You are simply having parathas, you are simply having burgers. So you are having carbohydrates like that. So carbohydrates, when they enter inside the body, they are broken into very simple molecules which we call monosaccharides, which we will be studying also. And these monosaccharides, when they enter inside, they are linked as isomers. And always remember one thing, ये जो हमारा ग्लूकोज है ना, ग्लूकोज इट इज द मेन हीरो, सबसे इंपॉर्टेंट हीरो है ये. अगर if we start cutting off carbohydrates, we can easily reduce weight because carbohydrates, excess of glucose is converted to form fats in the body. Okay, so aldoses, aldehyde as the functional group, ketosis, ketone as the functional group, and on the other hand, we classify depending upon the number of carbon atoms. So to begin with, always remember number of carbon atoms में carbohydrates always starts from a 3 carbon atom. So whenever it has 3 carbon atoms, we call it as triosis. Right? And aldoses, the example will be glycerol aldehyde. And ketosis example will be dihydroxyacetone. And again, the interesting part is beta, glycerol aldehyde and dihydroxyacetone are linked as isomers with respect to one another. Right. Similarly, if we talk about 4 carbon atoms, we call it as tetrosis. Right. Example of tetrosis is erythrose and erythrulose. If we talk about 5 carbon atom, it is pentosis. Example is, in aldoses, we can simply say ribose and ribulose. And if we talk about 6 carbon atoms में, the example is hexosis. Can anybody give me the example of hexosis? Very simple. It is glucose and fructose. Then there are 7 carbon atoms also. Can anybody give me the example of 7 carbon atom? It will be known as heptosis and example will be pseudoheptulose. So always remember all whatever names I have written, they are very, very important sources of energy. Simple, because why eat food? Why we eat food? Because food gives us energy, isn't it? So these are all the sources of, yes, very good Sayed, glucose and fructose are hexoses. An example of heptosis is pseudoheptulose. And it is very interesting also, there are certain derivatives of sugars also. Very important. Now, what are the derivatives of sugar? Like, if I take the example, I can say amino sugars. If we talk about amino sugars, we can simply say glucosamine. No, as such, it is not important. Yes, you are right, Sayed. The example of this is pseudoheptulose because this pseudoheptulose is a intermediate of HMP shunt. Because here we are focusing on those which plays a very, very important role in the body. So pseudoheptulose plays a very important role in the body being a intermediate of HMP shunt which gives energy. And yes, I just wanted to ask you people, beta, mode of communication is okay? Or shall I use mixture, English or Hindi both? Or English is fine with all of you? Is the mode of communication, I mean, you people are okay with English or shall I use both English and Hindi? So example of amino sugars is glucosamine is there, galactosamine is there. The only difference is what happens is amino group is added, so it will become an amino sugar. Okay, then sugar alcohols are also there. Sugar alcohols means sorbitol, which is a very important alcohol derivative, mannitol and galactitol. These all play a very, very important role in the body. Okay, so this is the classification of carbohydrates depending upon their functional groups. So we can say that they are divided into aldoses, ketoses, and these are the examples. Okay, mixture would be better? Fine. So wherever I'll see if you are unable to understand certain things, I'll explain that thing in Hindi. No issues. Okay. So triosis, this is the example. Tetrosis, 4 carbon atoms, these are the examples. Pentosis, these are the examples. Hexosis, these are the examples. And heptosis, these are the examples. Right. And this is how we classify. Then another way of classifying carbohydrates is depending upon the units. Because always remember, we call carbohydrates as saccharides. Why we call them as saccharides? Because they have been derived from sugarcane, which is Saccharum officinale. That's why they are saccharides. So we can say, if we talk about saccharides, simply it will be monosaccharides, right? Then it is disaccharides, oligosaccharides, and polysaccharides. So this is the classification as per their number of units. So if we talk about monosaccharides, first of all, monosaccharides में the most important examples are glucose, fructose, galactose, and mannose. These are the physiologically important sugars. Physiologically important means they are required by the body. Their metabolism is there inside the body. And if we talk about disaccharides, physiologically 3 disaccharides are there: lactose, maltose, sucrose, and trehalose. Yes, they are essential sugars also, because they are essentially required by the body. Coming over to oligosaccharides, monosaccharides have only one subunit, disaccharides have 2 subunits, oligosaccharides have 3 to 10 subunits, and more than 10 comes under polysaccharides. Right? Now, oligosaccharides and polysaccharides we will discuss after discussing mono and disaccharides. Now, first of all, we are going to discuss monosaccharides, their properties, their structures, and their biomedical importance. Clear? So, now see, monosaccharides, all of you are familiar with this. The most important property of monosaccharides is isomerism. So, if we talk about isomerism, we all know how do we define isomerism? Molecules having same molecular formula but different structural formula. Molecules having same molecular formula but different structural formula. Now, if we talk about the structure, for example, isomerism is actually of 3 main types. Right? First is your structural isomerism, which is a very simple isomerism, and it is clear from the word itself. Structural. Then second is stereoisomerism. And third is optical isomerism. Right? So if we talk about the structural isomerism, simple, you can say structure will be different, but molecular formula will be the same. For example, if we talk about glucose and fructose, right? ग्लूकोज एंड फ्रक्टोज का इफ वी सी मॉलिक्यूलर फॉर्मूला क्या है? इट इज C6H12O6, right? So glucose and fructose have same molecular formula as C6H12O6. But if we talk about the structures only, structure can be different as it is a aldohexose, right? And fructose is a ketohexose. Clear? Glucose aldohexose, fructose is a ketohexose. This is the difference, and this is how we classify the structural isomerism. Got my point? Is it clear? It's very simple. Coming over to stereoisomerism. Now, beta, if I talk about stereoisomerism, iso, same molecular formula, different structural formula, but different structural formula will be there in the space. So stereo comes from space. So difference in the spatial conformation, spatial arrangement, that will be there, and we will call them as stereoisomers. Now this you have to understand. Normally what we do is we always take glycerol aldehyde as a reference molecule. Okay, why? Because glycerol aldehyde has how many carbon atoms? Just now I told you it is a aldotriose. So how many carbon atoms it has? 3 carbon atoms, and this is the structure of glycerol aldehyde. CHOH, CH2. Clear? Now see beta, this is first carbon atom, this is second carbon atom, this is third carbon. So what inference we get from just seeing the structure of glycerol aldehyde? I can say that it is having a 3 carbon atom, and then it has a chiral carbon atom. And what do we understand by the this chiral carbon atom? That all the 4 substituents, if you look at the substituents, all are different. This, all are different. So how do we define a chiral carbon atom? A chiral carbon atom is defined as that carbon atom in which all the 4 substituents attached are different. Right. So if we are going to form a mirror image, obviously the mirror image will be non-superimposable, like OHH. It has a chiral carbon atom and it forms a non-superimposable mirror image. Clear? So this chiral carbon atom remains the same. Okay, so this inference we get from glycerol aldehyde. Now the concept here is that how do we assign a configuration? Just suppose if we have to classify, there are 10 students in the class, everybody will have a different name and we are going to assign them a different name. जैसे कई बार क्या होता है, टीचर्स भी क्या करते हैं, they assign a pet name to them. अब glycerol aldehyde है, we call glycerol aldehyde as D-glycerol and we call it as L-glycerol. So there has to be a specific aldehyde. So there has to be a specific nomenclature. Yeah, you can say we have to assign a standard nomenclature so that the higher number of sugars we can assign some group to it. ठीक है. अब this is the structure of glycerol aldehyde. Okay, and this is the structure of, if I say glucose. Glucose has this structure: CHO, CHOH. Because glucose is what? It is a aldohexose, so it has 6 carbon atoms. Clear? So 1, 2, 3, 4, 5 and 6. Okay, so this is the structure of glucose, which has 6 carbon atoms. अब glucose को just by seeing glycerol aldehyde, can we refer glucose as D or L form? No, because glucose has how many hydroxyl group? 1 group, 2 group, 3 group, 4 hydroxyl group. And if we talk about the chiral carbon atoms, glucose has 1, 2, 3 and 4. So it has 4 chiral carbon atoms. Right. अब now see how we have to name glucose. Glucose को भी कुछ नाम तो देना पड़ेगा. So what we do is we see this glycerol aldehyde. That is why I told you glycerol aldehyde is a reference molecule. Reference molecule in a sense that we say that if OH group is on the right hand side, right, we call it as D-glycerol aldehyde. And if OH group is on the left hand side, we call it as L-glycerol aldehyde. Clear? Okay, so I can simply say that in glycerol aldehyde, if OH group is on the right hand side, we will call it as D-glycerol aldehyde. And if OH group is on the left hand side, we will call it as L-glycerol aldehyde. Right? Now, just by looking at the structure of glucose, how we will name glucose. So normally what happens is this second carbon atom of glycerol aldehyde, okay, plays a second last carbon atom in the successive sugars. I'll just repeat it again, that the second carbon atom of glycerol aldehyde is going to behave as a second last carbon atom. Second last carbon atom कौन से? This is the carbon, right? So whether it is pentosis, whether it is heptosis, the second last carbon atom will always be given preference. If you look at second last carbon atom, OH group kahan pe? It is on the right hand side. So we will refer glucose as D-glucose. So this is the structure of D-glucose. Halki, glucose is going to form a non-superimposable mirror image also. That will be L-glucose. Right. But glucose is most stable in D form. So I can say that since glucose is most stable, yeah, hi, Mr. Ali, is most stable in D form, we call it as dextrose. And you know, in the hospitals also, you will never get glucose as D-glucose. You will always get glucose in the form of dextrose. So this is what is important. Just suppose, I'll just give you one common example. Just say, so many students are attending my class, isn't it? So say for 15 minutes, you are going to sit like this. Uske baad, if you feel uncomfortable, you will change your position. Okay, I cannot change my position. Yes, okay. But yes, you people have the liberty to change your position. So you tend to go in a stable conformation. Why you tend to go in a stable conformation? So that you can attend to the lecture more carefully. You can pay more attention, isn't it? Some of you prefer just lying down, lying down on the bed, carrying a mobile in the hand. Halki, that is a very bad habit. You should not read like that. You should always sit in a proper way, because whatever you are, reading. Yeah, hi Jia. Yeah. Okay. So basically what I'm trying to, yeah, good morning, Jia. So what I'm trying to tell you here is you always tend to go in a stable conformation. So most of the molecules also tend to go in a stable conformation. So glucose will always go, yeah, glucose will be always stable in D form. And that is why we always say that the other name of glucose is D-glucose. Very, very clear. This is what you have to understand here, right? Up now, Yeah, hi Vaibhav. Two Tiwaris have joined together. Okay, so it has four chiral carbon atoms. Now coming over to this, since they are non-superimposable mirror images of each other, they are known as enantiomers. So I can समझ नहीं आया जिया ओके मैं समझा देती हूँ देखो आई थिंक यू हैव आपने थोड़ा सा लेट क्लास को ज्वाइन किया है इजंट इट चलो फिर भी मैं थोड़ा बता देती हूँ अभी हम लोगों ने आइसोमेरिज्म स्टार्ट किया है और आइसोमेरिज्म में हमने किया है कि ग्लिसरल एल्डिहाइड एक एल्डो ट्रायोस है एल्डो मींस एल्डिहाइड इज द फंक्शनल ग्रुप ट्रायोस मींस उसमें 3 कार्बन एटम्स हैं ठीक है सो ये क्या करेगा OH ग्रुप आपको दिख रहा है OH ग्रुप कहाँ पे राइट हैंड साइड पे है तो जब OH ग्रुप राइट हैंड साइड पे होता है हम उसको कह देते हैं वी कॉल इट एज डीग्लिसरल एल्डिहाइड जब OH ग्रुप लेफ्ट हैंड साइड पे होता है उसको हम बोल देते हैं एल ग्लिसरल एल्डिहाइड ओके सो डी और एल दे आर लिंक्ड एज स्टीरियो आइसोमर्स स्टीरियो माने उनकी जो ओरिएंटेशन है वो डिफरेंट कहाँ पे वो स्पेस पे डिफरेंट है राइट एंड सबसे इंपॉर्टेंट है इट फॉर्म्स अ नॉन सुपर इम्पोजेबल मिरर इमेज क्लियर तो ग्लूकोज का भी यही एग्जाम्पल बताया कि ग्लूकोज में 6 कार्बन एटम्स होते हैं एंड 4 आर what 4 are the chiral carbon atoms 6 carbon atoms 4 are chiral carbon atoms chiral carbon atoms पता है जी या chiral carbon atoms वो होते हैं जिनमें all the 4 substituents attached are different ठीक है ये भी बहुत इंपॉर्टेंट है जिसमें all the 4 substituents attached are different और what I'm trying to tell you here is that glucose जो है glucose is most stable when it is in D form वो स्टेबल क्यों है डी फॉर्म में क्योंकि रीजन मैंने आपको बता दिया क्योंकि उसको क्या करना है उसको अपनी मैक्सिमम एक्टिविटी करनी है अब वो मैक्सिमम एक्टिविटी किसमें शो करता है व्हेन इट इज डी फॉर्म chiral yes but beta hybridization is never asked in the exam now whether it is sp3 sp2 it is not asked ओके यहाँ पे द ओनली क्वेश्चन विच इज आस्क्ड इज कि क्या होते हैं यहाँ पे वी आर नॉट टॉकिंग अबाउट हाइब्रिडाइजेशन कि वेदर इट इज sp3 और वेदर इट इज sp2 हियर वी विल बी फोकसिंग ऑन ओनली द पोजीशन ऑफ OH ग्रुप एंड फिफ्थ कार्बन एटम पे OH ग्रुप राइट हैंड साइड पे तो इट इज डी ग्लूकोज अगर फिफ्थ पोजीशन पे इट इज ऑन द लेफ्ट हैंड साइड इट विल बिकम L दिस इज फिफ्थ कार्बन लाइक दिस okay or glucose is more stable in dextrose so how do we define enantiomers enantiomers kya hote hain now you can easily define enantiomers okay isomers whose mirror images are non superimposable to each other and they are example D and L. Yes, these lecture notes, I can provide you with these lecture notes. What I'll do is, I have my group on Telegram. So what you can do is, I'll just post this PDF on my Telegram group. And after the class, after the lecture, I will provide you with my Telegram group also. So you will have latest information about the notes and whatever you require about biochemistry. Okay, my Telegram group is by my name only, Biochemistry by Pooja. I'll share the link with you people. Don't worry about that. Notes will be provided to you. Right. So isomers whose mirror images are non-superimposable to each other, example B and L. But beta, always remember capital D or capital L. It is never small d and small l. कभी भी छोटा small d और small l नहीं होगा. It will be capital D and L. Right? तो इनको हम ये भी बोल सकते हैं एक तो नॉन सुपर इम्पोजेबल टू ईच अदर DNL ठीक है और ग्लूकोज द अदर नेम ऑफ ग्लूकोज इज डेक्सट्रोज इज इट क्लियर Okay, now if I ask you one question, can you give me an example of aldohexose? Let me see. एक मुझे aldohexose का example, तो this is a question I am asking you, and give me the example of keto tribes. Can anybody give me the example of aldohexose? बेटा देखो इसको कैसे करना है. एल्डो मीन्स एल्डिहाइड. मैम हमको गुड मॉर्निंग का क्वेश्चन, जीएनएम का क्वेश्चन चाहिए प्लीज. जी आप नर्सिंग कर रहे हो बेटा? हाँ तो बेटा नर्सिंग में भी ये कवर होता है. इट्स कवर्ड इन नर्सिंग ऑल्सो, ऐसा कुछ नहीं है. देखो बेटा बायोकेमिस्ट्री, चाहे तुम लोग नर्सिंग पढ़ रहे हो, चाहे फिजियोथेरेपी पढ़ रहे हो, कोई भी कोर्स पढ़ रहे हो, सब में बायोकेमिस्ट्री आएगा. बट यस, नर्सिंग में थोड़ा ज्यादा न्यूट्रिशनल बायोकेमिस्ट्री आता है, बट दीज आर द बेसिक्स. जिया ये भी आता है. ठीक है. सो एल्डोहेक्सोस का एग्जांपल क्या हो जाएगा? या गुड मॉर्निंग रोश. ओके यस सैयद. कीटो ट्राइब्स का एग्जांपल. ओके बेटा जीएनएम भी इसमें कवर होता है जिया, ऐसा कुछ नहीं है. फिर भी इन केस इफ यू वांट, मतलब थोड़ा सा और गाइडेंस की जरूरत है, तो तुम मेरे से वैसे कनेक्ट कर लेना. आई विल हेल्प यू, डोंट वरी अबाउट दैट. बट ये सारा ऐसे ही आता है, क्योंकि बायोकेमिस्ट्री बेटा वो तो बायोकेमिस्ट्री ही रहेगी, चाहे किसी कोर्स में भी हो. ओके. हाँ कीटो ट्रायोस का क्या एग्जांपल हो जाएगा? वेरी गुड. देखो एल्डो मने एल्डिहाइड, हेक्सो मने 6 कार्बन एटम, तो इसमें तो आंखें बंद करके एग्जांपल विल बी ग्लूकोज. कीटो मने कीटो ग्रुप, ट्रायोस मने 3 कार्बन एटम, सो इसमें एग्जांपल विल बी डाइहाइड्रोक्सी एसीटोन. वेरी गुड. अजिया तुम मेरे से बेटा क्लास के बाद कनेक्ट कर लेना, मैं तुम्हारी हेल्प कर दूंगी, डोंट वरी अबाउट दैट. ठीक है. सो यस, सो अगर ऐसा क्वेश्चन आता है, फॉर एग्जांपल इफ दिस टाइप ऑफ क्वेश्चन है, यू शुड बी एबल टू सॉल्व. अगर सपोज इफ आई आस्क यू अबाउट कीटोहेप्टोस, फटाफट से बताओ व्हाट विल बी द एग्जांपल ऑफ कीटोहेप्टोस. एनीवन, व्हाट विल बी द एग्जांपल ऑफ कीटोहेप्टोस? बेटा कीटो कीटोनिक ग्रुप, हेप्टोस 7 कार्बन एटम्स, तो क्या हो जाएगा एग्जांपल? ये नहीं हो जाएगा सीडो हेप्ट्यूलोस? ठीक है, गेटिंग माय पॉइंट? आ रहा है ना समझ? यस वेरी गुड सैयद, गुड. दिस इज हाउ यू हैव टू आंसर. ठीक है, तो ये क्या हो गए? ये हो गए स्टीरियो आइसोमर्स. देन अनदर वेरी इंपॉर्टेंट क्वेश्चन is asked in the exam, that how to calculate the stereoisomers. How to calculate the number of stereoisomers. Okay, matlab I'll try to make biochemistry as simple as I can. Rest is in your hands, because the main duty of the teacher is to just deliver the lecture. Then when the ball goes in your court, you have to handle the ball, isn't it? If you strike it well, you will rise in life. Okay. So how to calculate the number of stereoisomers? There is the factor named want of factor. Okay, there was a scientist named Want of. He said 2 raised to power n is the formula to calculate the number of stereoisomers. N क्या है? N is the number of chiral carbon atoms. After number of chiral carbon atoms, beta Sayed, listen to my lectures, and you would be, after listening to my lectures, after understanding the entire biochemistry, you would be the first person to say, I mean, there are so many people who have said this, Again, you will say this ki biochemistry is the easiest subject. Why biochemistry is the easiest subject? Because you people have already studied chemistry. Biochemistry is what? Whatever is occurring in your body. Isn't it? If I talk about a chemical reaction, if you mix sugar and water, okay, I'll just give you one clear example. When you are mixing sugar and water, you are making a solution, you are making a homogeneous solution. Okay, keep on adding sugar, the solution will be saturated with sugar. उसके बाद क्या हो जाएगा? The water will not take sugar. सेम थिंग हैपेंस इन आवर बॉडी. हम ज्यादा कंज्यूम करते जाएंगे कार्बोहाइड्रेट को, नॉर्मल रहेंगे तो इट विल बी और ओके. बट इफ जैसे इन डायबिटीज मेलाइटस व्हाट हैपेंस, द कंसंट्रेशन ऑफ शुगर इंक्रीजेस because our body is now saturated, it cannot take in more of sugar. So what are the repercussions of that? What are the harmful effects of that you are going to study? Since you are going to become doctors, it is your duty how to make that solution back to homogeneous. Getting my point? So you can see how interesting it is. कोई भी रिएक्शन बाहर हो रही है, you increase the rate of a reaction by adding a catalyst. In the body, those reactions are catalyzed by enzymes. बस तो enzymes क्या हैं? They are the biocatalysts. Nowadays, so much of research is being carried on in the field of medicine. करते क्या हैं? What we are doing? We are synthesizing enzymes outside and we are trying to put them inside in a human being so that they can be expressed. That is what is your recombinant DNA technology. Right? We are recombining. So this is all mixed. So coming over to this, so how we calculate the number of stereoisomers? Want of factor is there, 2 raised to power n. N क्या बताता है? Number of chiral carbon atoms. So if we talk about glucose, how many chiral carbon atoms glucose has? It has 4 chiral carbon atoms. Right? So 2 raised to power 4 will be 16. So I can say glucose can form 8D and 8. So this will be the answer. Yes, basically it is the organic chemistry only. Yes, you are right. ठीक है. So glucose में कितने carbon atoms होंगे? 2 raised to power 4. It has 8D and 8L. Now can anybody tell me glycerol aldehyde has how many carbon atoms? Chiral carbon atoms? It has 1 chiral carbon atom. So it will be 2 raised to power 1 equals 2, which is equal to 1D and 1. Right? This is how we calculate the number of stereoisomers. Yes, very good. Right? So this is about stereoisomerism. Yes, very good. Now next isomerism is optical isomerism. Right? Now optical means isomers, so we know same molecular formula, different structural formula. Optical is their response to the plane of polarized light. Okay, so if I say, well, first of all I'll write the definition. Isomers which differ in a response to the plane of polarized light. Okay, and depending upon again on the chiral carbon atoms. So if I say this is a test tube which has a solution, right, and a plane of polarized light is passed through it, molecules हैं, atoms हैं, so it will rotate either in a right direction, and we study it with the help of a polarimeter. This is the polarimeter. Right? Okay, so whether it is going to rotate in right direction, whether it is going to rotate in left direction, right? If it rotates in right direction, we call it as dextro-rotatory. And if it is rotating in left direction, we call it as levo-rotatory. Like this. So dextro is denoted by plus and LIVO is denoted by negative. So positive is in the right direction, negative is in the left direction. But if it is rotating, it is optically active. When the molecule rotates, it is known as optically active. Okay, so whether it rotates in right direction, whether it rotates in left direction. So this is known as optically active substance. अब now if I ask you, if I mix the two, plus minus हमने mix कर दिया D और L, एक तो mixture बन गया. So it will be known as what? It will be known as a racemic mixture. Now what will happen to optical activity of this? Now you tell me, racemic mixture will be optically active or inactive? My question is, if a molecule is individually rotating the plane of polarized light, whether it is in right direction, whether it is in left direction, it is known as either dextrorotatory or levorotatory. My question is, if I mix the two, okay, plus or minus mix kar diya, what will happen to the optical activity? If I say it will be optically inactive, what do you say? Beta, it will be optically inactive. Why it will be inactive? Just because of one simple reason, that positive will cancel the negative. Because dextro will nullify the levo. So I can say that racemic mixture is, okay, so racemic mixture is optically inactive. But if I write small d and small l, say this is D, this is L, it is optically active. But when we mix, it is known as, yes, very good, internal compensation. Yes. So if you have studied chemistry, clearly you must have studied about mesotartarate and all. Internal compensation, external compensation, but यहाँ पे you don't have to deal with that. Don't worry. यहाँ पे question is generally asked, what is a racemic mixture? What is the optical activity of that? So always remember, racemic mixture is optically inactive. Okay. So these are optical isomers. Then we come over to, yes, then we come over to a very important, again, you can say isomers, they are known as anomers. Now, if we talk about anomers, now see, anomers are the isomers which differ at C1. So how do we define anomers? We defined as anomers are those isomers which differ at carbon 1, and carbon 1 is known as anomeric carbon atom. Right. Now see, I'll simply write the structure of glucose once again. We all know that glucose has this type of structure. CHO, CHOH, COH, third carbon atom, fourth carbon atom, fifth carbon atom, and sixth carbon. Right. Now, if we talk about the first carbon atom, I have written a differ at C1. Now what happens is, इसको हम क्या बोलते हैं? We call it as a open chain structure of glucose. Okay, or it is also known as Fischer projection. Just now I gave you the example, everything tends to go in a stable conformation. Agar suppose if you are listening to my lecture for since 1 hour, so automatically you will try to change your position, isn't it? So same is the case with glucose. Glucose, if it is existing in Fischer projection, it can change its position. So what happens is, position kahan pe change hogi? Along the functional group. So this CHO, we can write it like this also, CH double bond O, or it can be written like this, CH double bond O, because the resonance is there in the first position. now see, if you just imagine here, what happens is between the first carbon atom, now just pay attention and listen to it carefully. This is very important. Between the first carbon atom and fifth carbon atom, okay, there is loss of a water molecule. Means यहाँ से क्या होता है? It undergoes condensation. Up if double bond O is there, it will go from this side like this. And if double bond O is here, it will go from this side. So same is the case like this. Okay. So what happens is it is converted into this type of structure. Like HOH, HOH. Rest everything you see, it will be the same. This will represent now the first carbon atom, second carbon, third, fourth, and if we talk about here, opposite side में the reaction is there. So you see OHH, HOH, OHH, HOH, and CH2OH like this. So this remains the first carbon atom, second, third, fourth, fifth, and sixth. 2, 3, 4, 5, and 6. So can I say that the change is only at the first carbon atom? Yes or no? देखो बेटा, water molecule is lost. Okay, यहाँ से भी. So if I talk about this, if you simply say that either reaction is like this or reaction is like this. If it is loses a molecule of water from here, it will form this type of structure. If it loses a molecule from here, it will form this structure. If you see rest of the structure is same, second, third, fourth, fifth, and sixth are the same. So I can say that when hydroxyl group is placed downwards, this is placed downwards, we call it as alpha D glucose. Very important. But if hydroxyl group is placed upwards, we call it as beta D glucose. Right? Is it clear, beta? So I can say that if hydroxyl group is placed downwards, we will call it as alpha form. If hydroxyl group is placed upwards, we will call it as a beta form. So alpha and beta. बेटा क्लास का टाइम है 11 से 1, 11 to 1. Okay, so what happens here is, so I can say अगर hydroxyl group नीचे है, if hydroxyl group is placed downwards, it is alpha D glucose. If it is placed upwards, it is beta D glucose. But change कहाँ पे? Change is only at the first carbon atom, which we call as anomeric carbon atom. So I can simply say that anomers are those isomers which differ at C1. And if hydroxyl group is placed downwards, it is alpha form. Since glucose is stable in D form, so I can write alpha D glucose. And if hydroxyl group is placed upwards, it is beta D glucose. Right? So I can say that alpha and beta are linked as anomers. Right? And anomeric carbon atom is C1. Yes, beta. If you are late, try to be on time because the class will start every day at 11 o'clock till 1 o'clock. So what you can do is, because obviously there is a relation when we started with the lecture and in middle if you are joining, so you have to correlate the class. So what Vrush, what you can do is right now, so from where you have attended the class, just carry on with the class. After this, you can watch the class from the starting because it's already there. Okay, and don't worry, I will be taking your class for 2 hours, but in between I'll give you 5 minutes break where when you can refresh yourselves. Okay, so I will be giving break to you, so you don't have to worry about that part. Right, Prush? So we can say that anomeric carbon atom is C1. Now see if anomeric carbon atom is C1 and it is the functional group also. What is the characteristic of functional group? Functional group is the one which can reduce the reagents, isn't it? जैसे हम लोग Tollens reagent we study this Tollens and one more was there which you studied in your class 12. So aldehydes and ketones में this is the main difference. So if I say if the functional group is free, that means it can reduce, isn't it? So I can say that those sugars, this is very, very important beta, those sugars which have a free anomeric carbon atom, they are known as reducing sugars. Very important. Sugars which have a free anomeric carbon atom, they are known as reducing sugars. Clear? Yes, spellings. Yeah. So if I say any sugar who is reducing, just keep one thing in mind that it will have a free anomeric carbon atom. And there is one very important thing. Reducing sugars are the most important abnormal constituent of urine. Dekho, all of you know about urine. Since you are going to become doctors, isn't it? What is the role of biochemistry in the field of medicine? I'll just ask you one question. अब देखो, see, जैसे suppose now, whenever you have any problem, whether you have fever, whether anything, when you go and visit a doctor, doctor क्या बोलता है? Doctor will first examine you and doctor will tell you to go for the biochemical test, isn't it? अपना blood test करा के लाओ. So what is happening in those reports? You know, वो जो reports होती है ना, वो basically they are the खजाना. It is actually a very valuable treasure for a doctor. Doctor, after seeing the reports, will give you certain medicines. He will prescribe you something. So, same is, this is a very important one, detection of reducing sugars in urine. Yes, ये जो हमारा urine है ना, it plays a very important role in the diagnosis. What is the color of urine? What is the pH of urine? What is the, you can say, क्या कहते हैं उसको, कुछ भी होगा, एनीथिंग विच इज देयर इन यूरिन. जैसे ना यूरिन का कलर देखकर भी वी कैन सी कि मे बी दिस डिजीज इज देयर बाय सीइंग द स्पेसिफिक ग्रेविटी, बाय सीइंग द पीएच एंड बाय दीज एबनॉर्मल कॉन्स्टिट्यूएंट्स. तो हमारी बॉडी में देयर आर 3 एबनॉर्मल कॉन्स्टिट्यूएंट्स. Right. And what are these abnormal constituents? One is glucose itself. Right. All of you are familiar. एक होता है proteins also are there. And third is ketone bodies. This is a very good practical which you people are going to perform in first year also. So glucose, yes, urine analysis. Organic constituents, inorganic constituents, abnormal constituents, normal constituents, urine analysis is very important. तो जो glucose होता है ना, we normally see reducing sugars in urine because sometimes fructose also appears in urine, right? So to detect the abnormal, or you can say sugar in urine, the test we, yes, more commonly, yes, very good. Beta, it is known as glycosuria, and particularly if it is glucose, we call it as glucosuria. और कौन सा टेस्ट होता है? The test is your Benedict's test. Right. So this Benedict's test is carried out to check the presence of reducing sugars in urine. So I can say that अगर reducing sugar है तो उसका anomeric carbon atom क्या होना चाहिए? It should be free. And this Benedict's test, इसको हम बोलते क्या? This is also known as semi-quantitative test. It is very, very important. Okay, so what happens in this Benedict's test? इसमें होता क्या है? It has copper sulfate. It has a weak alkali. Generally, it has sodium carbonate. Generally, okay, and it has a salt. Sodium, potassium tartarate. These three are the main, you can say, ingredients of Benedict's reagent. Normally, Benedict's reagent is sometimes, it comes in the form of Benedict's reagent only. So what you do is simply you take urine sample and you add Benedict's reagent into it. So basically, what is the chemistry behind that? ये जो copper sulfate होता है ना बेटा, copper sulfate will dissociate. So it will dissociate into cupric and sulfate ions. अब ये जो क्यूप्रिक आयंस होती है ना, एंड व्हाट हैपेंस इज यूरिन में व्हाट इज देयर? यूरिन में रिड्यूसिंग शुगर इज देयर. So these reducing sugars have a tendency to form enidiols, right? And ये जो enidiols होते हैं, they are very good reducing agents. Right, so they are very good reducing agents, and ये जो weak alkali है, it is making enidiols. मतलब you have to just understand this, making enidiols. अब now what will happen is, we have, this is the urine sample. The moment we add Benedict's reagent into it, what will happen? Copper sulfate will be dissociated to form cupric ions and sulfate ions. On the other hand, whatever amount of reducing sugar is there in the urine, it will form enidiols, and these enidiols then will combine with this salt, sodium potassium tartarate, and in the end, these cupric ions will be converted to cuprous ions, and it will give red precipitates. Okay, now I have written that it is a semi-quantitative test, because depending upon the color of the sample, because what we are doing is, this is urine of the patient and we are adding Benedict's reagent. Okay, and then we heat it. So after heating, if the coloration is like this, gyre means the coloration can be green, the coloration can be yellow, it can be orange, it can be red. ऐसा तो नहीं है सारे डायबिटिक आएंगे, isn't it? कोई ना कोई नॉन डायबिटिक भी होगा, and by just looking at the color you have to give your prediction. So if it is green, chalo we'll start from red. If the coloration is red, we say the amount of sugar is 1.5, or you can say chalo more than 2%. We'll put it like that, more than 2%. If it is orange, then we will say 1.5 to 2.0%. If it is yellow, 1.0 to 1.5%, and if it is green, 1.5 to 0%. 0.0 like this. Okay, so if the coloration is green, so obviously he is not diabetic. But if it is orangish to red, he is going towards diabetes. Got my point? So अगर if I show you two solutions, say this is one solution, this is second solution, okay, and if the coloration is this, and if the coloration is this. Okay, so difference क्या है दोनों में? This is positive Benedict's test and this is negative Benedict's test because this is the giving the color of Benedict's test only. What my point? So ऐसे भी question आता है कि if two solutions are given to you and you have to analyze कि which of one of them is diabetic, which is not. So you will say इसमें red coloration है, he is going towards diabetes and he should be you can say prescribed to undergo thorough blood examination. And if it is green or yellow, that means traces of sugar is there. On which sugar? Reducing sugars. And that is why Benedict's test is known as what? It is known as a semi-quantitative test. Right? Why? Because just by looking at the colors, we can predict the amount of sugar. Is it clear to all of you? क्लियर हुआ बेटा? I hope it is clear. Jia, Vrush, Sayed, is it clear? Okay, so this is Benedict's test. I hope it is clear to all of you. Okay, right. So अब देखो, I'll just give you a gist of what all we studied. सबसे पहले we started with isomerism. Right, I told you isomerism या isomers are of three types. एक होते हैं structural isomers, एक होते हैं stereoisomers, एक होते हैं optical isomers. If we talk about the main one, जैसे structural, simple glucose and fructose. If we talk about stereoisomers are capital D and capital L, and they are also known as enantiomers. And optical isomers, which rotate the plane of polarized light either in right direction or in left direction. So either they are dextrorotatory or they are levorotatory. Then we came over to the most important one, the anomers. Anomers are the isomers which are differing only at C1. And since they are differing at C1 or the anomeric carbon atom, if they are free, I just repeat this term again, if they are free, that means they are reducing sugars. Why they are reducing? They can reduce. Whether it is Fehling solution, whether it is Benedict's, whether it is Tollens, any reagent. Why? Because of one simple reason, their functional group is free. And always remember, beta, all sugars are reducing except sucrose and trehalose. I'll show you the structure also, then you can closely, differentiate between the two. So this is reducing sugar, and you can say that reducing sugars ka there is only one test, which is Benedict's test. Fehling test bhi hota hai, but nowadays we don't perform Fehling test. Why we don't perform Fehling test? Because if you remember, Fehling test mein na strong alkali is there, sodium hydroxide. So normally what happens is enolization is weaker in strong alkalis. Got my point? यहाँ पे तो weak alkali है, so more enolization will be there. That means whatever reducing sugar is present, it will be detected easily. And this is the test. Okay, so these are anomers, and alpha and beta form of glucose are linked as anomers. Then second important term here is epimers. Right? So how do we define epimers? Anomers differ at C1. Epimers are the isomers which differ at any other position except C. So isomers which differ at any other position except C1 will be epimers. For example, galactose is a C4 epimer of glucose. Now see beta, if I talk about glucose, this is the structure of glucose. 1, 2, 3, 4, 5 and 6. Okay, so 1, 2, 3, 4, 5 and 6. Okay, so this is glucose and you can say कि first position पे it is there. Now if you see fourth position, hydroxyl group is placed downwards. If I write the structure of galactose, right? First carbon atom will be the same, second will be the same, third will be the same, but there will be a difference in the fourth position. Like this. Okay, so I can say that galactose is a C4 epimer of glucose. If glucose has hydroxyl group placed downwards at fourth position, galactose has fourth position पे OH up. Clear? Similarly, if I talk about mannose, I'll just write once again, see. I'll highlight it like this. See. Up now, you tell me galactose is a reducing sugar or a non-reducing sugar. Now I'm going to ask you. My question is whether galactose is a reducing sugar or not. Anyone? My question is galactose क्या है बेटा, reducing है या non-reducing है? Galactose is a reducing sugar. That means galactose can come in alpha form and we can write galactose in beta form. Okay, so what will be the structure of alpha D-galactose? Okay, so what will be the structure of alpha D-galactose and beta D-galactose? So if I talk about alpha D-galactose, you see, सबसे पहले galactose है, so galactose will be this. This is the fourth carbon. Right. Now coming over to alpha and beta, if I talk about alpha, this will be the structure of alpha and this will be the structure of beta D-galactose. Rest everything will be the same. HCH2 like this. So this is how we say that reducing sugars have alpha and beta form, and this is how they differ in the structure. Okay, no problem beta. Rush, if there is a network issue now, what you can do is you can just go through the lecture again offline also. I mean like once the class is over, because this will be always there on YouTube. And next day when you are coming for the class, so you can always ask me if there is any confusion. That is why I am going at a normal pace. I am not going very fast. I am not going very slow. I just want to keep in pace with you guys so that you guys don't face any problem. Okay. And notes definitely will be shared with you. So you don't worry. Notes are not running anywhere. I am not running anywhere. When I am there, I want to give you the best. Right. So we can say that galactose will always be a reducing sugar. Because reducing sugar is the one which is actually differing only at the first carbon. Because most of the students get confused between the structures. Even though structures are not asked, but since some of you are first prof students, first prof the structures can be asked. Okay. Then second example is of mannose. So mannose is what? Is a C2 epimer of glucose. So what happens is, if I write the structure of mannose, it will have this type of structure. Second position pe it will have OH here. First carbon atom will remain the same. Fourth will remain the same as that of glucose, like this. Right. So this will be mannose. Second position. Glucose का structure तो I have already told you. This will be glucose. Third, fourth, like this. So this is glucose, or this is alpha D glucose, and this is alpha D mannose. Right? Yeah, little bit, because see, these are very simple structures, because I told you, if you are going to give your first prof exam, automatically this will be very important, and little bit you should know better. I'm not going into that much detail, but yes, slowly, and this is since you are in class, you are in first prof, no? Right now it is easier for you to learn. And honestly, I'm telling you, if you know the chemistry, biochemistry will be very easy for you. And this is not too tough, because already you people have studied in your class 12. Okay, so this is mannose. Now, can you write the alpha D and beta D of mannose? I think now you can write. अब तुम लोग लिख सकते हो, I hope. ठीक है? So this is the difference between epimers and anomers. Okay, so if I have to just summarize again, anomers are the ones which are differing at C1. Epimers are the ones which are differing at any other position except C1. Okay, anomers are having, you can say, first free anomeric carbon atom, then they are known as reducing sugars. On the other hand, these epimers are also reducing sugars because their first carbon atom is free, but they will differ at fourth position if it is galactose, and it will differ at second position if it is mannose. So till now, all epimers, all anomers will be reducing sugars. Okay, now reducing sugars, I told you, they are the abnormal constituents of a very important abnormal constituents of urine, which can be detected by Benedict's test. Then another important property is that reducing sugars undergo mutarotation and OSA zone formation. Reducing sugars can undergo mutarotation and OSA zone formation. Now, first of all, what is this mutarotation? Muta mane itself. Rotation is a rotation. So, tendency to rotate within itself is mutarotation. So, when you can say tendency to to rotate within itself and is only around anomeric carbon atom. This is very important. ये होगा कहाँ पे? It will be only around this anomeric carbon atom. जैसे for example, if I write the open chain structure of glucose, that I've already written, I will say, or Fischer projection. This Fischer projection can be converted into alpha D glucose and it can be converted into beta D glucose. Right? Yeah, that is why my pleasure, Vrush. That is why I am just going through the basics. It will be very beneficial for you in the later stages. And it will make biochemistry totally crystal clear to all of you. And the only thing is that you also have to practice a little bit, because when you are preparing for your USMLE, when you are preparing for other exams, this is the one which actually student forgets, because first prof ke baad, once you start going to the clinics, once you start, you start forgetting the basic subjects. And if you have studied them thoroughly, then automatically they will be clear to all of you. So this is my advice to you as a teacher. You have to go through the basics. Okay, so open chain is the Fischer form, alpha D glucose, or it can be converted into beta D glucose, or it will give an angle of rotation 52.5 degrees, and this will be plus 19 degrees. Right? So I can say that they are rotating here and here, so it is rotating. So you can say characteristic of only anomeric or reducing sugars. Clear? Then we come over to the second property, OSA zone formation. Now see, if we talk about OSA zones, OSA zones are basically the crystals, because every molecule has its own melting point, right? So what we do is we simply, and you know that it has this aldehyde group, right? Or you can say it has a ketonic group. So we can say that aldehydes and ketones, since they have a free functional group, they will combine with phenylhydrazine. This you don't have to learn, that is why I am not going. Phenylhydrazine hydrochloride plus anhydrous sodium acetate. Okay, and you have to keep this in a water bath overnight. Right? So automatically glucose, fructose, all those reducing sugars, when they are kept in water bath and they are treated with this, they are going to form crystals. And these crystals, you have to take them out, okay, and you have to just put them on this slide like this, and you have to observe them under the microscope. So if you have this type of structures like this, needle-like. So if needle-like crystals appear, we call them as the crystals of glucose and fructose. Or you can say glucosa zone or fructosa zone. Right? If crystals are thorny edge-like, then the crystals are of galactose. You will see all are reducing sugars. Right? And if crystals are powdery puff, it will be of lactose. And if they are sunflower petals, like this, sunflower petals are like this, powdery puff is like this, right? And thorny edge like this, pointed structures, then it will be maltose. Okay, so this is OSA zone formation. OSA zone formation is simply characteristic of reducing sugars. What is happening is these reducing sugars have a tendency to combine with phenyl hydrazine hydrochloride and anhydrous sodium acetate, and they are forming crystals, and we put these crystals on a microscope. So if needle-like crystals are there, glucose fructose; if thorny edge, galactose; powdery puff, lactose; sulfaplatals, maltose. So always remember mutarotation and OSA zone formation is a characteristic feature of only and only reducing sugars. Right? Then after OSA zone formation, then we come over to another property of carbohydrates, which is oxidation. Now see, if I talk about glucose, okay, glucose has this structure 1, 2, 3, 4, 5, and 6. Okay, now we know that it has a aldehyde, it has a alcoholic group. So if glucose undergoes oxidation in the presence of mild agents, okay, bromine water hai, you can say this nitric acid is there, only this group will be oxidized. We all know that, जैसे when you studied in chemistry the conversion, or you can say ascent of series. So aldehyde to सबसे पहले alcohol to aldehyde to carboxylic acid, ऐसा ही होता है ना. So इधर क्या है, aldehyde is there, alcoholic group is there, so this will be oxidized, it will form aldonic acid or gluconic acid. If in the presence of strong oxidizing agents, both will be oxidized and it will form glucaric acid. Glucaric acid. But the most important one is when this is not oxidized, this is oxidized. That is possible only with the help of enzymes, and this takes place in the body. And we get a very important intermediate which is known as glucouronic acid. Now, most of the students get confused between gluconic and glucouronic acid. Okay, the most important difference is that here uronic acid is there. Getting my point? And enzymes. This reaction takes place in the presence of enzymes, and there is a very important pathway in our body जिसको बोलते हैं uronic acid pathway. And you know, uronic acid pathway, why this pathway is operative? Just to form glucouronic acid. Just to make glucouronic acid in the body, this is made possible in the body. Getting my point? And what is the significance of this? Why we have to make it in the body? क्योंकि देखो, whenever we are making something inside the body, it has to be beneficial for the body. Otherwise, there is no point. So, what is the significance of glucouronic acid? यहाँ पे the everything will be the same. Only here COOH will be there. This will be the one which will be oxidized. Rest will be the same as glucose. First carbon atom will be the same. Right? So significance is that it is required for the degradation of heme. What is heme? We all know hemoglobin is the main respiratory pigment in the body, which is responsible for the transport of oxygen from head to toe. And you know, this heme is synthesized also in the body. And always remember one thing. Yes, very good. It is a protein, hemoglobin. It is a chromoprotein basically. And always remember one thing. Whatever is produced in the body, it has to be degraded. That is what you are going to learn. कोई भी चीज हमारी बॉडी में बनती है वो डिग्रेड होती ही होती है. इफ इट इज नॉट डिग्रेडेड it will lead to diseases. If it is not produced, it will again lead to diseases. So hemoglobin, once its function is over, it is degraded. और degrade किस में होता है? Heme plus globin. Globin, which is giving globular structure, और heme is degraded to form bile pigments. तो जो glucouronic acid होता है, this is produced in the liver. Produced in the liver and helps in degradation of heme by forming urobilinogen and stracobilinogen, which we will be covering in heme synthesis. Okay, so this is one very important point about इसीलिए glucouronic acid बनता है body में. क्यों बनता है? Because it is required for heme degradation. Then after this, these are the 2 points, and then it is required, this glucouronic acid is required for the synthesis of mucopolysaccharides, which again we will be discussing. देखो, if we talk about science, half of the definition is clear from the word itself. Mucopolysaccharide. Muco means wherever mucus is required, that viscous fluid is there, glucouronic acid will be there as one of the main components. Okay, and what I am trying to explain you here is glucouronic acid is produced as a result of oxidation by the body by uronic acid pathway. Clear? So this is how it undergoes oxidation. Then after oxidation, second property is reduction. Now, if we talk about reduction, always remember aldehyde, it will be reduced to alcohol. So I can say that during reduction, the alcohol will be produced. Sugar alcohols will be produced. For example, if I talk about glucose and if we talk about galactose and if we talk about fructose, these are the 3 very important sugars. So glucose in the presence of enzyme aldol reductase. Glucose in the presence of aldol reductase forms sorbitol. Galactose in the presence of again same enzyme aldol reductase will form galactitol. Okay, Jia, I will do that. You can contact me after this class, right? Galactose will form. Okay, sure, Jia. I've listened to your request. We will help you. Don't worry. So galactose will form galactitol, and fructose simply it will undergo, reduction. It will form again sorbitol. Right. Now, what is the difference between this sorbitol and this sorbitol? Okay. See, first of all, this is produced from glucose, this is produced from fructose. So this sorbitol will give energy to the sperms. Okay. Coming over to this sorbitol, this is very important. Right. Now what happens is this sorbitol gives energy to the eye muscles. Because I told you, glucose is the main hero. Whatever amount of energy we require, it comes from the oxidation of glucose. So this glucose will undergo again, when it goes to the eye, because our muscles, eye muscles are blinking, contracting. So many issues are occurring in the eye also. Energy, from where we get the energy? We get the energy from glucose. So glucose forms sorbitol, which gives energy to the eye muscles. Ab, what is the clinical correlation of this? जैसे you must have heard बेटा कि whenever we talk about diabetes, isn't it? Diabetes is a killer disease. The worst effect of diabetes is on the eye. Okay. The person can become blind also in diabetes. Why? Because what happens is in diabetes mellitus, simple you can say, the concentration of glucose is more. It becomes more. So if the concentration of glucose becomes more in the eye, the concentration of sorbitol will also become more. Sorbitol will also become high. So this sorbitol, problem with sorbitol is it is unable to go outside the eye. Why? Because it is impermeable to the eye membrane. Impermeable to the eye membrane. Okay. Now what happens is if it is impermeable to the eye membrane, say suppose this is the eye membrane and this is the increased concentration of sorbitol. Okay, these are the vessels like this. So if sorbitol cannot go out, it will start exerting pressure on the cell membrane. So उससे क्या हो जाएगा? It will increase the ocular pressure. Right. So just compare it with the help of a balloon. Yes, I'm coming to that. Yes, very good, very good. Retinal degeneration occurs in diabetes. What is the biochemical basis behind that? Biochemical basis is this, we call it as retinopathy, pathological disorder in retina. So when the concentration of this increases, it starts exerting pressure on the walls. So उससे क्या हो जाएगा? It's like a balloon. So it will burst. Bursting से क्या हो जाएगा? There will be bleeding from the eye muscles. So automatically there will be a defect in the eye. So it leads to increased ocular pressure, which leads to the formation of cataract. So what type of cataract? We call it as a snowflakey cataract. Snow means, this is snowy. Whenever you go in snow, that hazy thing starts appearing. Same is the case in diabetes mellitus. The person, when the concentration of sorbitol has increased, so cataract, snowflakey cataract appears. और इसका treatment क्या होता है? This is the symbol of treatment, RX. करते क्या है? Aldol reductase inhibitors are given. Aldol reductase inhibitors are given क्योंकि we cannot reverse in the eye. Once diabetic retinopathy is there, we cannot reverse it, but we can control it. How we can control it? By obviously controlling sugar. Second is aldol reductase inhibitors are given, which will not let the concentration of sorbitol to increase. Okay, so what is sorbitol? It is the increased, you can say it is the sugar alcohol which is produced by aldol reductase enzyme in the presence of glucose. Clear? And if we talk about galactose forms galactitol. And what is this galactitol? Again, same, increased concentration of galactose. Right, we will study this disorder. This disorder is known as galactosemia. Vrush, I didn't get you. Enlist what? So galactose will give galactosemia. Once we discuss the metabolism of this, then we will discuss. Don't worry, everything will be discussed, everything will be covered. Okay. So in galactosemia, what happens? Again, galactose ka concentration increases. So it will lead to increased concentration of galactitol, which is also known as dulcitol. Right? So what will happen? Again, it will lead to the formation of cataract, but this cataract is known as bilateral cataract, right? So this is due to galactosemia. I will discuss each and every thing once we will discuss the metabolism. Right now I'm just telling you the basic structures which are produced. So which that will also be discussed later. Right. So this is what is galactosemia, or this is what is the reduced product of galactose. Clear? And then comes fructose. Fructose with the reductase is there. It gives sorbitol, which gives energy to the sperms. So here up till now we finish with the basic chemistry, and just 2 days. Tomorrow we will be finishing up with the chemistry of carbohydrates. So this is bilateral cataract. So today what we discussed was, I'll just give you the summary. We discussed about the characteristics of monosaccharides. So we discussed about isomerism. We discussed about anomers. We discussed about epimers. We discussed about oxidation and reduction. And obviously, the most important one we discussed about was reducing sugars. So here we finish up with the characteristics of monosaccharides. Tomorrow we will be finishing up with the entire chemistry of carbohydrates. And this material beta I will be sharing in my Telegram group. So what I'm thinking is, once I finish up with the entire chemistry of carbohydrates, then only there is some point sharing it. Otherwise, half of the part will be shared today, half of the part will be shared tomorrow. So again, it might create a confusion. So tomorrow I will be uploading this material. Okay. So I hope till now it is clear to all of you. Any confusion, any problem in structure, in whatever topic? I'm also eager that you people should attend, because it is a continuity beta, so automatically you will understand. Okay. And rest we will discuss tomorrow. So if you don't have any confusion, we can wind up the session for today. So see you guys tomorrow sharp at 11 p.m. tomorrow. Okay. So have a great day. See you guys tomorrow. And one more thing before ending up the class, beta, whatever confusions you have, no, suppose if you have gone through this lecture, no, so try to make your notes also. But this is an advice to you as a teacher. Start cultivating a habit of making your own notes. No doubt, the teacher is going to share the notes with you, but when you have the manuscript with you, whatever information you get from other sources, you can just add it to that. So it will be very much helpful to you. And Jia, if you have any problem, you can contact me after this, and I am ready to counsel you also. No issues. So guys, have a great day. See you guys tomorrow. Till then, take care. Bye bye.